general transcription factor, binding to the TAT box and saying, hey, RNA polymerase, there's a gene here. All right. Activator proteins can enhance the function of TF2D. So let's say, here's our gene, here's our core promoter. TF2D is binding to the TAT box here. Our activator is nearby binding to an enhancer, and it's also interacting with a co-activator, which is helping TF2D recognize the TAT box. But TF2D is more likely to be able to come into this promoter because our activator is here. Okay, so that's one way an activator could work. All right. Now repressors, on the other hand, remember they bind to silencers? The way repressors can work is they can actually grab TF2D and prevent it from doing its job. So they inhibit TF2D from finding the TAT box. And if TF2D doesn't find the TAT box, right, RNA polymerase can't come in there and bind to the core promoter. So that is going to, of course, result in the silencing of transcription. So over here, transcription is going to be off. Over here, transcription is going to be on. So these are some ways that regulatory transcription factors can interact with TF2D. Now remember we said mediator mediates regulatory transcription factor interaction with RNA polymerase. Okay, mediator is a giant complex of proteins, which we're just depicting as this big red blob. This big red blob consists of a lot of different proteins that you don't need to know the names of. Okay, over here, far away, is a DNA element. In this case, it's an enhancer. Our activator protein binds to this enhancer. Okay, and this activator is going to interact with mediator. And when it interacts with mediator, mediator is going to help RNA polymerase switch from initiation to elongation. So it's going to help turn transcription on. Over here, we've got a silencer bound to a repressor protein interacting with mediator. All right. This is actually repressing the switch to elongation. Mediator is actually holding RNA polymerase at the transcription start and preventing it from switching to elongation because of this repressor. Okay, so some repressors are going to inhibit mediator, some activators are going to stimulate mediator and help RNA polymerase move into the elongation phase. So that is going to upregulate transcription. Yeah?
Can you ever have like an activator protein bind to a silencer or repressor bind to an enhancer?
So these sequences, right, are specific DNA sequences, and these proteins have specific shapes, and they're only going to recognize their specific regions that match, right? So that would be very unlikely to happen. Okay. So now that you have the basics of how regulatory transcription factors work, we need to talk a little bit about how regulatory transcription factors themselves might get regulated. All right? Like, how does the cell tell the regulatory transcription factors what to do? Okay? So nucleotide cells, you should be getting the picture that there are levels upon levels of regulation, right? Because we look at this picture of mediator, and I told you, even though we're only looking at one enhancer or one silencer, there could actually be multiple enhancers or multiple silencers or a mixture of silencers and enhancers for any one gene, right? So you could have multiple levels of regulation. The regulatory transcription factors themselves can be regulated. So one way is by small effector molecules. So things like hormones can bind to these regulatory transcription factors, change their shape, and then cause them to bind to their silencer or enhancer region, depending on what kind of regulatory transcription factor they are. Or they could be regulated by a protein-protein interaction, such that they have to bind to each other before they become active. Or our regulatory transcription factor might have to be modified by another protein and be phosphorylated, so a post-translational modification, addition of a phosphate group that activates it before it could do anything to activate or repress transcription. So even the transcriptional regulators are regulated, okay? So there are lots of factors that are going to play a role in whether a gene is stimulated to be transcribed or whether it is repressed. Okay, question so far? Are your heads going to explode? Tell me. Yes? 6 seconds. Breathe in, breathe out. I know, right? Don't you wish? You wish. Yeah.
Are we going to go over the regulations for the the hormones?
So, I mean, basically, when you think about hormonal regulation, that is actually one of the more simple things to understand, because either the hormone is present or not present. If it's present, right, it can bind the transcription factor. The transcription factor is going to change its shape, and it's going to go bind the thing. Right? So that's one of the more simple ones to understand. Like, in my mind, phosphorylation is one of the harder ones to understand, even though it's just the addition of a phosphate group, because there's usually a big long chain of events that lead up to that phosphorylation. And that's harder to understand because there might have been 15 different events that led up to this phosphorylation, right? Okay, but the whole point of what I've just been saying so far, right, is that these regulatory transcription factors bind to the enhancers or the silencers. They exert their effect either through TF2D or through mediator, right? And they're either going to upregulate or downregulate transcription, depending on whether they're activators or repressors. Okay? So really the picture is pretty straightforward. But remember, we're talking about eukaryotes. And how is eukaryotic DNA packaged? Remember? Nucleosomes. Really tightly packaged, what do you think is going to happen to gene expression? What do you think? Really tightly packaged chromatin, isn't that going to prevent RNA polymerase from getting in there and transcribing? So we need to think about what's going to happen if the gene needs to be transcribed, but it's in some tightly packaged chromatin. And what's going to happen is this thing called chromatin remodeling. All right? And this can create either what we call an open conformation or a closed conformation. And these are exactly what they sound like. Open means more loose, hence more transcription. Closed means tighter, that would be less transcription. Okay? And a lot of this process depends on energy. You need ATP to modify chromatin. You're going to need proteins to help with this process. They're going to use ATP. And they are going to reposition the nucleosomes. So you got a bunch of nucleosomes, and then they're moved around or spaced out. Or completely removing a nucleosome. You got a region full of nucleosomes, and one of them gets removed. Or change in the composition of the nucleosomes. Histone proteins are actually members of protein families. Each member of the family has a different role in chromatin compaction. So you've got standard histones in these nucleosomes. A chromatin remodeling complex can come in and replace a nucleosome with a histone variant nucleosome. And that's going to affect how tight this DNA is packaged, which we'll talk about in just a second. So these are your three big categories of what can happen when we're talking about remodeling chromatin. We're talking about changing the position of the nucleosomes. We're talking about removing a nucleosome. Or we're talking about replacing some histones in a nucleosome to change how that nucleosome is packaged. Yeah.
Is this similar to like the part where we learned about heterochromatin and euchromatin?
Yes. So most genes that are expressed are not in really tightly packaged heterochromatin, yet still, if they are packaged in nucleosomes, this is going to inhibit the ability of RNA polymerase to move through and transcribe. So some amount of chromatin remodeling is always going to be needed to transcribe a gene, even if it's not in heterochromatin. Okay. So, we've talked about the composition of nucleosomes. They have H1, H2A, H2B, H3, and H4, right? And H1, remember, is the linker. And then there's two copies of H2A, H2B, H3, and H4 in the nucleosome. But these proteins are encoded by genes that are parts of gene families. And there are many different versions of these histones. The human genome alone has 70 different kinds of histone genes. And some of these different histone genes play special roles in chromatin structure and function. So for example, at the centromere, there's a special histone H3 called SENH3. And this is essential for creating the kinetochore. Go back here. There's another one, let's see, what is it, H3.3. This is a histone that helps promote a more open conformation of the chromatin. And you'll find this particular H3 at genes that are expressed. Okay, so this is what we're talking about when we're talking about replacement of histones with different histones. At expressed genes, often you're going to find H3.3 instead of H3. Another thing that happens to our histones to promote expression or silencing is histone modification. the three major modifications of histone proteins are acetylation, methylation, and phosphorylation. And the reason why these modifications are important is they actually alter how tightly the DNA is bound in the nucleosome. And that can influence whether or not RNA polymerase can get in there and transcribe it. So we're looking at our 4 core histones here. And each of these histones has this long tail part of it that's not part of the core kind of globular structure. This is where we find a lot of the modifications. When you look at the nucleosome, the tail kind of switches. Alright? Acetylation, a lot of acetylation on the tails tends to be a modification that causes the DNA to be less tightly bound around the histones. So when we talk about chromatin modifying complexes, some of the chromatin modifying complexes are coming in here and actually doing things like adding acyl groups to the histones. And that is causing the DNA to be less tightly bound. And that's going to allow RNA polymerase to get in there and interact with it. All right. So, the tightness of the DNA wound around the nucleosomes can be influenced by, what do you guys think? Post-translational modification of histones, replacement of histones with histone variants, nucleosome positioning, or both 1 and 2. All right, I see a lot of 4s. All right, is the position of the nucleosome going to actually affect how tightly the DNA is wound on the nucleosome? Isn't the position of the nucleosome just moving the beads on the string? It's not changing the tightness, right? Just moving the beads. The tightness, right, is affected by whether or not your histones are, say, acetylated, or, right, whether or not you've replaced H3 with H3.3. Right, those things are going to give you more loose wrapping of your DNA around your nucleosome. The positioning of the nucleosomes is just moving the nucleosomes around on the DNA. The tightness is staying the same though, right? So best answer here is going to be 1 and 2 is actually affecting the tightness of the DNA wound around the nucleosome. All right, so let's talk a little bit more about positioning. So we get a better picture. All right. So we're looking at a transcriptional start site, the +1, right? So here's the promoter, here's the gene, this is our transcriptional terminator. It turns out that if you look at where the nucleosomes are in a bunch of genes in a eukaryote, you will see that there tends to be a region right around the core promoter that does not have a nucleosome. And there are 2 nucleosomes that we call the -1 and +1 nucleosome right around that region that are marking where transcription is going to start at +1. It's as if, right, the promoter is kind of saving a spot for RNA polymerase to start transcription. and there's no nucleosome there. But for RNA polymerase to transcribe through this region, something's going to have to happen with these nucleosomes. All right. So transcriptional activation, a lot of stuff is going to happen here. All right. We kind of waved our hands earlier and said that our activator is going to help activate transcription. So now we're going to actually see what an activator might be doing. So here's our transcriptional start site. Here's an enhancer sequence. An activator binds to the enhancer. It doesn't have to be right next to our transcriptional start site. We're just showing it here because it's easier for us to see. The activator protein, once it's bound here, it can actually bring in other proteins that are going to do stuff. Many times what these other proteins are going to do is they are going to do things to these nucleosomes. They are going to carry out chromatin remodeling. And that is really what is going to help RNA polymerase switch from just initiating transcription here to actually elongating. So in this case, our activator brings in a chromatin remodeling complex, which is a really common one called SWI/SNP. And SWI/SNP brings in a histone acetyl transferase. That is going to acetylate the tails of the nearby histones. Remember, that's a modification that causes the DNA to loosen its grip on the nucleosome. Alright, so DNA is loosening its grip, pre-initiation complex starts to form, and then we can switch to elongation. Alright, and so what's going to happen is as RNA polymerase moves through here, these nucleosomes might get evicted and might have to reform behind RNA polymerase. So when the DNA loosens its grip and RNA polymerase moves through, as RNA polymerase moves through, those nucleosomes might lose their binding completely from the DNA. And then they might have to be completely replaced. So you see evicted histone proteins getting replaced here. All right, so our activator is actually activating transcription by helping to bring in chromatin remodelers. And that's what's allowing RNA polymerase to be able to transcribe.